This video describes the results of Startup Project ER20-1127, the development of a time-integrative passive sampler designed to measure perfluoro and polyfluoroalkyl substances in water. A passive sampler is a device that's placed into the environment, like this flowing water stream. As the water goes by, the sampler will absorb substances from the water, much like flypaper capturing insects. But instead, these chemical contaminants are then captured and returned to the lab, where they can be analyzed to understand the water chemistry. Our passive sampler design is shown here. The device is relatively small and constructed of polyethylene, with an absorbent designed to capture perfluorinated compounds, or PFAS compounds. The blue color absorbent is in direct contact with the water as it's held between mesh screens. The absorbent is specifically designed to absorb PFAS. First, it has a weak base ion exchange group resin, which is augmented by copper ions to enhance short-chain PFAS binding. By the way, copper is also useful as it's biocidal and prevents biofilm growth. The resin is also hydrophobic to enhance the binding of long-chain PFAS compounds. It should be noted that there are mesh screens on both sides of the passive sampler, allowing good contact between the absorbent and the water. Some of the key features of the device is that, number one, it's relatively small, so it's easy to deploy and conceal. Second of all, it has a very high capacity for PFAS compounds, so it can absorb compounds in an integrative fashion over the period of several months. Thirdly, it's pre-wetted, so there's no conditioning field steps in the field. It's simply removed from the package and placed into the water. Finally, it's easy to ship. Its small size means it fits in a standard 50 ml Falcon tube that can be placed on ice and returned to the lab for analysis. Let's see how that works. After a sample is returned from the field, there are 5 steps in the analysis. The first step is to rinse it off, and then the second step is to place it in a surrogate solution. Surrogates are isotopically labeled standards of the PFAS compounds that are to be measured. Those are bound in a short duration of time, and then they are desorbed along with the PFAS analytes into methanol. That methanol is evaporated and reconstituted, and then run on HPLC mass spec for the quantitation of the different PFAS analytes. In the first step, the passive sampler is rinsed. This is to remove any dirt or debris that might have accumulated during the sampling in the natural water. To remove any of the DI water or other residues, it's handy just to put it into a centrifuge tube again and then centrifuge it to remove the extra water and any debris. Afterwards, it is then removed and then placed in an aqueous solution containing the surrogates that are used for the quantitation of the PFAS analytes. This is also handy for QA/QC parameters. The surrogate solution with the samplers is then shaken for several hours to quantitatively bind the surrogates. We found that this takes about 8 hours to get complete absorption of the surrogates. Residual surrogate solution is removed by another step of centrifugation to spin off any residual liquid prior to the next step of the analysis. The sampler is then removed and then placed in a methanol solution to desorb the surrogates and the analytes. The next step is to then shake it in the methanol solution for a few hours to desorb the analytes, and they're ready for reconstitution, which is evaporated under nitrogen, and after it's evaporated, they'll be reconstituted in an internal standard solution and run on LCMS. That's the procedure. What the liquid chromatography mass spectrometry actually measures is the mass absorbed to the passive sampler. This is the amount of PFAS compound that had been accumulated during the sampling time while the sampler was in the water. Now what we really want to know is the aqueous phase concentration in the water. To do that, the mass of the PFAS compound that's been measured is divided by the length of time that it was in the water and a constant. That is the sampling rate, which has been measured for these samplers under given circumstances. The user will be given a lookup table for these RS values. So after measuring the mass absorbed, take the time, divide it by the constant, and that returns the aqueous water phase concentration. I hope it is evident that it's easy to deploy these samplers, analyze them in the lab, and then calculate the aqueous phase concentration as shown here. Again, one of the main advantages of a passive sampler is that it gives a time-weighted average for the PFAS levels during the duration of the sampling event. This is very handy if there is a site that has variable concentrations of PFAS over a period of time. Such a site would necessitate multiple grab samples during that time period. This can be replaced with a single passive sampler. We expect that these passive samplers will be another useful tool in the toolbox for site monitoring in order to return important PFAS concentration data across many different contexts. Let's look at some of the results that we obtained during our CERTIP-funded project. One of the sites we measured at was Peterson Air Force Base located in Colorado. Passive samplers were deployed in groundwater monitoring wells for 1 to 2 weeks at the site. In addition to collecting the passive sampler results, grab samples were also obtained during the site investigative work for comparison. Here's the data that was returned from the project. What this is that the concentration of PFAS measured in the grab sampling results is plotted against the PFAS concentration measured against passive sampling results. If there's a correlation between them, they'll fall on the 1-to-1 line, which is the diagonal across the plot. A range of both short-chain and long-chain PFAS compounds were measured during this part of the project. What we found is good correlation between passive sampling and grab sampling results at Peterson Air Force Base. One important thing to note is that there's good correlation across over 5 orders of magnitude of PFAS concentration. This indicates that passive samplers should be effective in a wide variety of concentration environments. If you want to learn more, please reach out to the personnel listed on the website about ER 20-1127. Any of us will be happy to work with you and answer your questions about passive sampling for PFAS. I hope you found this video informative. Thank you for watching.